This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 525, recorded on December 14th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How's it been?
Great.
Also,
end of the semester.
And that's right.
Yeah.
Is everything done, or are finals done?
No. Students are starting finals, or are in finals. Yeah, I don't pay too much attention.
It varies because our younger son is on his way home right now. He's done. His finals this week. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Our finals done there. Who knows?
Yeah, I'm taking the day off. Actually, there was a Facebook post from my daughter's university of the graduation ceremony. I didn't even know that was happening. So they're graduating. And she just sent me a text that's I got to read to you. Hang on. Wait for it. She says, just read this. Quote, I don't know where she got this. If you took all your DNA, straightened it out, put it end to end, it would stretch to Jupiter and back 10 times.
Wow.
You are neither small nor insignificant. You're just very well folded.
Yeah, that's good. I like that. What is it from? I don't
know. I will text her back and see if I can find the source.
Well, I learned yesterday that the surface area of the average adult lungs is equal to that of a tennis court.
Ooh.
That sounds about right. I think I've heard that.
It's not as good as Jupiter, but that's pretty big. Yeah. Also joining us from Madison, New Jersey, Brittany Barker.
Hi, it's great to be here this week.
Are you done with finals?
I think the last finals were today. I am very much in the midst of grading finals grading final papers.
All right. Well, keep it up during the show.
Oh, okay.
From Western Massachusetts, Alan Dove.
Good to be here. And the only end of semester information I have is my daughter got her report card recently, and she was bummed out that she got an A- in math.
What grade are we talking about here?
This is 7th grade.
7th grade.
Yeah, she was really aiming for that A+.
Good.
Yep.
Well, keep trying.
Yeah.
And we have a guest today for this episode. But we are back to Bozeman, Montana. Our guest is Michelle Flenniken. Welcome to TWiV.
Thank you. Hi, everybody.
Montana
Hello.
State University. And you are in the Department of Plant Sciences, Plant Pathology. You're also co-director of Montana State University's Pollinator Health Center. It's an interesting job.
Yeah.
I'm sure we'll hear about it. Well, I've been thinking about having you for some time. So here's our chance to talk about honeybees and their insect and their viruses. And we follow up on an episode this week by Raul Nandino, which I released midweek and probably freaked everyone out.
Yeah, they probably don't know what day it is now.
Yeah.
So I was really confused by the message on my phone that there was a new episode.
Yeah, as we discussed last time, I just didn't know where to. We had no Fridays where we weren't going to record for some time, so I just released it. And in fact, I have a bunch of episodes that are going to happen like that over the next few weeks. One just happened today. Do any of you know the name Peter Hotz?
Yes.
Yeah.
So apparently he's visiting today, and I got a text from his host saying, Peter would like to do a short TWiV on his new book.
Oh.
So, and his book is going to be my pick of the week, so I won't tell you about it, but so we recorded the shortest TWiV ever, 20 minutes.
Oh.
Wow.
Oh.
Which is basically an advertisement for his book.
First couple of episodes were like 30 minutes or something, right?
Really?
I think so. They're really, really short. It's just you and Dixon talking about West Nile virus and
So that's why you decided to join, because you figured they were going to be short, right?
Yeah, just, Friday afternoon, sure, I can spare half an hour for a podcast.
I wanted to make an announcement. The American Society for Virology program for the annual meeting in July 2019 has been announced. It's going to be at the University of Minnesota in Minneapolis. And I want to make a couple of points. One is that travel awards will be available to members, and dependent care awards will be available to members. So now it's only December 15th. The abstract deadline is February 1st. The deadline for the travel awards is February 4th. And for now, we don't have a deadline for the dependent care awards. But because of those upcoming deadlines, now would be a really good time to become a member or pay your dues. So you just go to the ASV site, asv.org, choose membership, and sign up. We just, as of today, put the poster on the homepage of the ASV site. It's also in the mail to all of you, and the program itself will soon be on the Minnesota website.
I found that email very exciting yesterday.
Yes. Yeah.
So, Michelle, do you, I wanted to ask you, do you ever run into David Quammen?
I do, actually. I heard you guys on TWiV not that long ago, and David talked about brainstorming titles with scientists, and I was in one of those brainstorming sessions. And yeah, I run into him sometimes.
At the local supermarket or something?
Yeah. I guess a brainstorming station was at a place called Montana Aleworks, and my husband, Blake Wiedenhoff, sometimes golfs with him. So we run into each other from time to time.
I guess Bozeman's not so big, right?
No, I think it's about 35,000 or 40,000 people.
Oh, wow.
All right, so let's start with a little bit of your history. Michelle, where are you from originally?
I'm from Iowa originally, right next to Iowa State University in a little town called Nevada, and spelled like Nevada.
Ah, that's confusing.
Yeah.
Which everyone pronounces Nevada, right?
We all say Nevada, but if you're not from there.
Yeah. So you went to school through high school there, I guess?
Yep, I went through high school there, and then I went to the University of Iowa for undergrad studies. But I actually started school in Sinoche, France. So my parents moved to France when I was little, and I started school there.
Oh, cool. So you speak French now?
I used to. I've in my later years, I focused in on trying to learn Spanish. I think it's more useful. And so now I have kind of like, I think, one center of my brain that has a few different languages, none of them very solidified. And so I can say a mixture of things.
How long were you in France?
3.5 years.
And then, what ages?
Through kindergarten, I started first grade again back in Iowa.
Okay.
Oh. What part of France were you in?
It was a little town called Sinoche, and it's about 2 hours southwest of Paris.
Yeah. It's a nice country, right?
Yeah, beautiful.
Were you a science major at Iowa?
Yeah, I was a biology major and a chemistry minor, and I also did a humanities minor as well.
Oh.
Were either of your parents scientists?
My mom is a computer programmer, and my dad is an engineer.
Ah.
Okay. What kind of engineering?
Agricultural engineer. That's what brought us to France, actually. He worked for John Deere, and so he was transferred there from Iowa to France for a few years.
Okay.
Tractors.
Yeah. That's right.
Make good tractors, I understand. I never owned one.
Yeah, neither have I.
There's a bit of a thing going on now with John Deere. Apparently they have some disputes with people who would like the right to repair their tractors.
You're not supposed to repair them? Yeah.
Yeah, their newer tractors are apparently supposed to be serviced only by John Deere, and that's raised some hackles.
Michelle, what got you interested in science? Can you remember back where the interest came from?
Yeah, I was thinking about that actually in preparation for this show, and I would say I was always good at science and math in school. And then a fifth grade science teacher, actually, he brought individuals on field trips, and he brought me to the Veterinary Research Center at Iowa State University in fifth grade. So I was exposed to scientific research then, and I think it was really influential. It's pretty cool that he did that. He was his first year of teaching when he was doing that. His name was Pat Sullivan, or is Pat Sullivan, I should say.
When you went to college, did do you already have in mind that you want to get a PhD after that?
No, actually, I was deciding at that time, I would say my town, although close to Iowa State University, was this pretty small town, and I didn't think of being a scientist as a job, so I was kind of thinking I'd go to medical school. And so I finished school, took the MCAT. I actually moved to Bozeman, Montana, right after undergrad to work in a lab. I worked in labs all throughout undergrad. And then I joined the Peace Corps and went to Ghana for 2 years after taking the GREs and the MCATs. And I was deciding whether I should be a doctor or a scientist while being a forestry volunteer in the Peace Corps. And, somebody would come to a kid in the neighborhood or something would get a bloody nose and I would almost pass out. And then that pretty much solidified that I should become a scientist.
So were these labs that you worked in through college and then in Bozeman, what kind of things were you doing?
Yeah, first I did drought tolerance research in Arabidopsis. And then here in Bozeman, I studied a fungal pathogen. In high school, actually, I did the Women in Science and Engineering program and also worked in a plant pathology lab.
Mm.
Did you ever have any training in entomology?
I didn't actually. When I was in Raul Andino's lab as a postdoc, that's kind of when honeybees were dying, and I was in a virology lab that also studied RNA viruses, and a lot of the viruses we'll talk about today are like polio in the Picornaviridae family. And then RNAi, of course, was just a big news then, and we knew we were starting to appreciate that RNA interference was important for Drosophila antiviral defense. And so I wondered about bees, and Raul and I drove to UC Davis, where I started to learn about honeybees right away, and I kind of changed my attention to studying honeybees in around 2008.
That's actually where I first met you, in Raul's lab. I had gone there to record Risky Business with Raul Andino, and I met you.
Yeah, that's right.
And you said, I think you were about to go off to your job, and you said you were going to work on honeybees. So that planted the idea of having you on TWiV. And this is many years later now, because that was many episodes ago, I think, for that one.
So did we skip over some stuff here?
Yeah, we did.
Raul was a postdoc, right? No, it's okay. Raul was a postdoc, right? So, we left you in Ghana.
Yes, so
Right.
he left me in Ghana. So in 2000, I came back from Ghana, and this is kind of interesting too. I applied to graduate school by candlelight and by mail. I don't think anybody does that anymore. Where I lived in Ghana had no electricity. And so they kind of knew who I was because, of course, the administrative assistants weren't getting many of these type of applications. And so I came to Montana in 2000, Montana State University, where I did my PhD in microbiology with a combination of advisors, a virologist, Dr. Mark Young, who studies the viruses that infect the archaea that live in the hot springs of Yellowstone National Park and other cool places in the world.
Or hot places.
What? Really
hot places.
That's right. I should say hot, but really fascinating. And then a chemist, Dr. Trevor Douglas. And so I worked with both of them, and I think that was a great training for my PhD.
Mm. So Mark Young has always promised to take me into the backwoods of Yellowstone and show me all the cool things.
The hot things now.
Yeah, hot things. Yeah, never happened, but it would be a cool TWiV episode walking around doing that. Maybe one day.
And
Yellowstone's amazing.
That for sure. He guest lectures in the virology class now, and we had our virology final last night actually. And so we can definitely, next time you're in Montana visiting, we can definitely take you to Yellowstone National Park.
Is there, Kathy, is there a plan for an ASV? I don't think so, right, in Bozeman?
It's not on the books. No, not yet.
Just get it on.
We can lean on Michelle.
So we can do it with Yellowstone. Yeah, you're at that point in your career now where you could start thinking about.
And I remember thinking about it and thinking that MSU, I need to look at the holding capacity here, and the ASV has gotten so huge lately, so we can accommodate everyone here.
Yeah, it might be.
You've got a lot of wide open spaces.
Yeah, it's true. When they did have ASV here, the socials and things were outside under the stars in a tent.
Mm-hmm.
We did that.
It was nice.
I remember that. Yeah, it was very nice. I remember you going off to run one afternoon. And
yeah, I'm a runner.
Yeah, you and Raul, and he's like, "You want to come with us?" And here I am in my clothes and pants, and shoes. So, yeah, sure, I'm ready.
Yeah.
So you then you went from your postdoc right to a faculty position there at Bozeman, right?
Yes. Mm-hmm.
Nice. You don't have to travel far, right?
No.
All right, so let's talk about honeybees. So you said you got interested in Raul's lab because of the colony collapse, right?
Yeah.
So tell us about honeybees, why we should care, and what's colony collapse and all that.
Yeah, so honeybees are really fascinating eusocial insects. They live in colonies of up to around 40,000 individuals, and primarily those are all female workers that are sterile. And then there's one queen bee, and she's the famous one, but I always tell the kids that I visit with that she's not, as her name suggests, the ruler of the colony, but her sole responsibility is laying eggs, and she lays about 1,000 eggs a day. And so the colony, it's really fascinating to study bees because you can study them as a superorganism or as individuals. And so we do both in my lab, and I found that very interesting. But backing up a step, why bees are important. So honeybees are primary pollinators of many of the crops that produce the foods we enjoy, such as fruit, nuts, and vegetable crops, as well as pollinating numerous plant species that enhance the biodiversity of all landscapes. Interestingly, though, honeybees are not native to the U.S. They were brought here by the European settlers in the late 1600s. And so although we rely on them a lot in the U.S., for pollinating crops and things, they're a non-native species.
Where are they from?
Then again, so are the crops.
That's true. That's a really good point. I usually say that as well. So they're from Europe originally. Of course, I mean, it depends when you want to go, or Africa, originally. Right. It depends how far back we go. But now they're responsible for pollinating. They estimate, every third bite of food you take is influenced by a honeybee pollinator, because of course even things like alfalfa, which is used to feed cows, for those of you that are carnivores and eat cows, you still need alfalfa to feed the cows. And so a lot of things require pollinators.
How long does a queen live?
So queens can live a few years, and actually that's one of the things that beekeepers are most concerned with. When I go out and talk to beekeepers, they're saying their queens don't live as long as they used to, and that's a big concern. And you mentioned colony collapse disorder, and that was first described kind of in 2006 or 2007, which is about the time that I was getting interested in investigating the viruses that infect bees. But colony collapse disorder, of course, TWiV listeners will appreciate that it's a disorder because we don't know what cause, if there is a single cause of colony collapse disorder. And it's really a definition of an absence of a lot of things. It's an absence of known bacterial pathogens, high mite infestation. The queen is still there often, and there's enough food stores, but the colony has died. And so now they term this colony collapse disorder, but you can probably guess already that it's pretty hard to diagnose. And today we think that it's only responsible for a small percentage of overall colony losses, which average 33% annually. Colony collapse disorder is probably only responsible for about 5% of those losses. So when I talk to groups, I usually say I study, honeybee colony losses or deaths, but not specifically colony collapse disorder.
Okay.
We used to have a beekeeper listening way back in 2009. Right. Tom and Suki Glenn of Glen Apiaries in California. They used to write, and he's told us he used to listen while doing something with his queen, or with something or other. But
we may have other beekeepers listening. They just haven't
there was a student whose name escapes me right now, who actually went on to do a postdoc at NIH, who kept bees as a hobby.
So, Michelle, the Glen Apiaries?
Yeah, I've met them before. They did queen rearing, and so he might have been grafting queen cells when he was listening. I don't remember.
That's right. That's right.
Yes, that sounds familiar.
So
okay, Rich and I are on the same line and talking at the same time. That's going to be good. So 2006 is when colony collapse disorder was described, and that also seems like that's a significant year when hive losses and commercial apiaries went up from reading your papers, right?
Yeah, and I think one of the things that really grabbed my attention early on is that anecdotally it was described, by beekeepers who had been keeping bees for generations. And so they noticed something in the field that was dramatically different than what they've seen before, and that they would describe kind of waves of death in their apiaries or bee yards where, maybe 100 or so colonies are stored. And so they would say that it was kind of going through the colonies. And so it really did sound, from those types of description that a pathogen was involved. And so that's what really piqued my attention as a microbiologist.
Okay. But this inflection point, so one of the papers you mentioned that hive losses are, and you just, I think, mentioned that hive losses are about 33% in these commercial pollination operations. But historically, they were in the 10 to 12% range.
That's correct.
What happened? That's
the big question. So that's the big question. And so, what has changed? What's different? In the late 1980s, this mite called the Varroa destructor was introduced from Asia into the United States, and that really decimated feral colonies. We call them feral colonies because really they're kind of escapees from some managed honeybee populations, and then now they're living in hollowed out trees or other locations, not in these boxes that you typically associate with managed honeybee colonies. But that mite really decimated those feral colonies, and also beekeepers took a big hit in the late 80s due to this mite. And then what are the other factors, and so we often say that these losses are multifactorial. And of course, we say that when there isn't just one thing impacting bee health. And is it increased agrochemical exposure or particular agrochemicals, limited floral resources or inadequate nutrition, different beekeeping management practices, which include, that over 60% of the managed bees in the U.S. are transported on flatbed trucks to the Central Valley of California around Thanksgiving, actually, to be placed there prior to the almond bloom, which occurs each February. So then this is a big difference in management. And then, of course, pathogens play a big role. And it's thought that this combination of this Varroa destructor mite and then the viruses that it vectors may be a substantial reason impacting higher numbers of colony losses. And again, I mentioned also poor queen health. Why are queens living fewer years than they used to? And so these are really big questions, and there isn't just one answer. There's, of course, much research and a lot of findings in, kind of single cohorts, but not one universal factor has come up throughout all studies.
Do you think there's a role for glyphosate in the collapse as well? That's been in the news lately, right?
So I saw that, and I think Nancy Moran's group has a recent paper in PNAS. Right. So glyphosate, I would say its biggest problem with Roundup and glyphosate is that it reduces floral diversity. If the more of that we're using, the less flowers for bee nutrition that are available. And so increased use of that, certainly in the Midwest, has really reduced available forage. And so that's probably the most major impact that it has on bees. And then, that recent study indicates that it may impact the microbiome of the bee, and that may in turn impact microbial infections as well. But I think its biggest factor has been reduced forage.
So the bees get trucked around to pollinate the almonds. Don't they get trucked to the Northeast to do blueberries?
Yeah, some of them do. Mm-hmm.
And so
probably not the same colonies that go to California.
Right.
Though, right?
Yeah. The Florida colonies probably go up to do the blueberries. There's kind of like an eastern loop and then a western loop. And then some of the Florida colonies come to California as well.
I'm so glad that none of them go for the cherry blossoms.
They come to Montana, where we have cherries in the northwest of Montana.
Does that industry of trucking them around, does that correlate to some extent with the loss of colonies?
Yeah, that's an interesting question, and one I wondered about myself when I first got into this. But as I mentioned, often beekeeping operations are multi-generational, and they'll tell you that transportation of bee colonies today is vastly improved over what it was, when their grandfathers did it, for example. And so we have better trucks; they're better organized. They even have bee-specific forklifts that lift up pallets of bees of 4 colonies each, and they load the truck at night and they drive. I've seen this where they give the truck driver a certain route, so they're driving all the time, and so the wind keeps the bees in, and then they unload the colonies in California at 4 a.m. And so they say they've gotten better at transportation. And the first longitudinal monitoring project of colonies that I was involved with, I was surprised to find out that the pathogen signature from colonies that were sitting right next to each other was often different. And we did not observe a spike in pathogen load after transportation events, provided they went well, provided the truck driver really got there at 4 a.m. and not 4 p.m. I've seen it both ways. The 4 p.m. is much more stressful for the bees.
Why does this have to be done as opposed to having local bee populations? Is it
Yeah, that's an excellent question. And so really, when you think about February in the Central Valley of California, there's really nothing blooming. And many of those native and wild bees, which many of them are ground nesting bees, actually are still in the ground, and it's prior to their emergence. And so honeybees are, since we can transport them in these Langstroth colonies, those white boxes with removable frames, and they're pretty tolerant and adaptable to that with our large-scale mono agriculture, and a big bloom that only lasts a couple weeks in February, really, in order to pollinate that, the only way to do it is to transport these managed bees into that area at that time. And
because I guess otherwise you'd have the hives sitting around all year and they wouldn't be productive enough.
Well, there wouldn't be food for them there.
Right.
Yeah.
They would be, and actually almonds require honeybee pollination. In a bee, you don't get any almonds. And so really it's there's 2 industries are highly linked. Almond growers and beekeepers have to work together.
Then after the season's over, the bees stay in that area long term?
Nope. Then they get trucked up for a lot of the ones that are from Montana, maybe go to Washington State to do apples, and blueberries there.
I can see the conversation among the bees. Hey, you coming on the apple drive?
Exactly.
We're going to go do apples now.
Yeah. And then they come back to Montana or South Dakota or North Dakota to forage on primarily alfalfa and wildflowers and things during the summer months. So really, they often at least have 2 stops or 3 stops.
So they're on tour all year round.
They're on tour. Yep. We call them migratory beekeeping operations.
They need T-shirts,
Yeah.
2018 bee tour.
They're migratory, not migrant. That's important.
Yes. And then also, I often get this question asked, and that we already talked about that they're trucked on flatbed trucks to drive them there. But sometimes I forget to say that part, and people ask me if they fly to all these places, and they don't. Bees fly around 2 miles.
Do you lose bees on the road at all?
They sometimes do. I think we've all seen those images online or on the news where, one of these trucks unfortunately has a accident, and then bees are everywhere, and they call the fire department to hose them down. And so, that happens every year. But
I'm sorry to laugh. That's a problem.
Yes, it's a bummer. But that's typically only 250 colonies, and we have about 2.5 million honeybee colonies in the U.S.
Wow.
And so that means over 1.6 million of them go to California. So, a truck unfortunately turning over once or twice a season isn't why we're having 33% annual colony losses.
But just
losses in transportation of 33% on U.S. highways would be a serious problem for a lot of reasons.
Yes, that's true.
But just driving, the colonies or the hives are covered in a way that the bees don't just fly out, right?
Sometimes, yeah. For long distance transport, they usually put a mesh over them.
I see.
Because they need to breathe. And that's it.
I assume that there are also colonies that stay put. They don't get moved around, right?
There are some, and our colonies here at Montana State University stay put all year. We actually put these things called bee cozies around them, keep them a little bit warmer.
I love it.
But we do that because we only have 10 colonies. But beekeepers who stay here all year, who are real beekeepers, they wouldn't do that. They might pile up some hay bales to block the wind, but they're not going to be pampering them as much as we do here at MSU.
Okay.
Are the losses similar in the transported hives and the hives and the stationary hives?
That's an excellent question, Ellen. And a lot of people have tried to look at that. And in some studies, we call those stationary operations. And sometimes you lose more in the stationary operation than you do in the migratory operation. And sometimes it's vice versa. And there's no universal theme there. And then I find those studies are just often confounded because when you think about what were those colonies exposed to, from a virus standpoint? The ones that stayed, let's say, in Minnesota or Montana versus the ones that went to California just have a totally different pathogen exposure, total different climate they were exposed to, forage. And so then so much is variable that it makes comparisons really difficult.
Right.
Do the bees get confused with all this travel?
So that's interesting too. They orient visually. So when they fly out of their colony, they're actually looking, and they fly out one way and then come back that same way. And so they don't get confused. They're pretty good at finding their directions.
Okay,
when they first arrive in California, they don't fly out and say, "Whoa, where the hell are we?"
Well, so yeah, they do, and this is pretty cool actually, and it's fun to watch this. When they first fly out, provided they got there at 4 a.m. and then got to fly out, slowly, what you'll see them do is leave the entrance of the colony that's at the bottom and do a few loops in front of the colony. You can actually see clusters of them doing loops and kind of getting a sense, and then they take off. Then they go. But they do take what they call an orientation flight to make sure they can find their way back.
And what are they looking for? Do they sense something, flowers and so forth?
So a lot of what they're actually looking for, it's cool. You can look at it on Google Images. How does a bee see? And it's different than we see. We think about these beautiful purple flowers that they might land on. They don't see the purple, but they see them kind of as landing pads. So flowers attract bees differently. And then they're looking, for floral resources, nectar and pollen. And then there's the really cool thing about bees is that they come back and then they communicate to their half sisters that there's a floral resource by this cool dance called the waggle dance.
Cool.
Right.
So they're placed so that when they're traveling, they're placed right next to whatever crop that they have to pollinate, I presume, not too far away, right?
Sometimes. So they're first dropped off in these kind of holding yards, and then they're next placed in the crop.
Mm-hmm.
So
neat. Well, let's talk a little bit about honeybee viruses. When were the first ones identified?
I think the first honeybee viruses were identified in the early 1900s. First by doing some just crude virus preps, and then try to reinfect bees. So these historic experiments in the maybe up until the 1950s, where they're doing these crude extracts, observing symptomatic bees, making virus preps and then reinfecting bees and describing symptoms. And then, of course, when we had transmission electron microscopy in the late 1930s, then these viruses could be then imaged, or more often than not, they were detected by antibodies that were specific to the viruses, and that's how they diagnosed them then.
And then with next generation sequencing, we could then look at their sequences, right?
Yeah, and so that, I think, really has exploded the number of bee-associated viruses that have been discovered. And then, of course, and as you guys often talk about on TWiV, then it's really important then to go back and then characterize those viruses. Many of them, as I mentioned before, are positive sense single-stranded RNA viruses. So about 30 nanometers in diameter. So you often get co-purification of several viruses. And that can be a problem if you're using those crude viral extracts, or even viral extracts at all, to do infection studies. And so some of the common bee viruses include the dicystroviruses, black queen cell virus, Israeli acute paralysis virus, Kashmir bee virus. Then there's the IFLA viruses, deform wing virus, sac brood virus. You can tell a lot of these are named after the symptoms that they were originally discovered. And then there are taxonomically unclassified viruses, including the Lake Sinai viruses, which I co-discovered with a graduate student at UCSF and with Joe DeRisi's lab there in San Francisco.
Great names.
Mm-hmm.
Yeah.
Yeah.
So can you have to grow these on bees? Is that right?
Yeah, and often what people do is infect larvae or pupa in the laboratory setting. And in general, you can get pupa that are relatively free of confounding infections or natural infections. And then you can introduce, let's say Deform wing virus or Israeli acute paralysis virus into these pupa, have them basically propagate the virus in the lab, and then you do a viral purification to isolate them. But this brings up an important point that there's no reliable honeybee cell culture or method for propagating virus. And there's only a single molecular clone, and that's a molecular clone of Deform wing virus by Benjamin Lamp's group in Austria that, I think the paper just came out a couple years ago. So honeybee virology, we have some of these tools like next generation sequencing, but we're also, I would say, doing a lot of old school virology because we don't have many of the tools.
Well, this brings up another issue, I think, too, I guess, that it's almost like another level of model organism to real situation. Then if you're infecting a single bee in the lab, that's not really the organism you're studying, right?
Yeah. So as what we study in the lab, I say we have 50% or less of our lab studies the superorganism, thinking about the whole colony, and then you focus on studies to look at the individual because you have to. There's too much variation in that individual colony, and then we even do experiments on hemocytes. So a PhD student in the lab, Alex McMenamin, isolates hemocytes, the immune cells of bees, from honeybee larvae in the summer. And then he can infect those with viruses and study things at the cellular level, which, of course, we really need to do. But we do all that with primary cells.
Okay. So I guess you could infect a bee and put it back in the hive, but then you'd have a biocontainment problem because it would just infect all the other hives, right?
You might, and also a tracking problem. And so you can put little trackers on those viruses, on the bees. And then you can also paint the bees. And this is how they figured out, they decoded or deconstructed the waggle dance. And they figured out that it's telling both directionality and, a distance of that food source by actually, painstakingly labeling bees with little numbers and tracking them. But so typically what we do is we do kind of correlation epidemiologic work when we're working at the colony level, and we always team up with commercial beekeepers for that type of data. And then when we're doing mechanistic studies about how does a bee fend off viruses, we do that at the individual bee level, and we bring those bees into the lab. We give them what we call artificial mite bites, a dose of a specific virus, whether that's a bee virus like Deform wing virus or Lake Sinai virus, or a model virus, which we often use Symbis tagged with GFP so we can track it. And then we know for sure that the bees didn't have this virus. And those bees are never let back out of the lab. We're a BSL-2 lab, and we keep those bees here, study them, and they sacrifice their lives for science.
Right.
So of these viruses that you mentioned, are any of them honeybee specific, or do most infect other things? And if so, what are they?
Oh, yeah, that's an excellent question. And so I think we often think and get mammalian-centric in our view of virology. And most of these viruses infect a range of insects. And so, for example, Deform wing virus was first discovered in honeybees, but infects bumblebees and probably over 20 species of bees. There are an estimated 4,000 species of bees in North America, hundreds of species in Montana. And really, we have only begun to study native and wild bees like bumblebees and halictid bees. So most of the bee viruses we know about get named that they're honeybee viruses, but they can infect ants and wasps and mites, those parasites that infect them. And so they're more promiscuous viruses in the insect world than we think about in mammalian virology.
Do some even infect plants?
So that's interesting. There was a paper a few years ago now that indicated by negative strand PCR that this tobacco ring spot virus actually replicates in honeybees. And of course, tobacco ring spot virus is a plant virus. And so that's kind of an open question. And that's probably the best and one example of that. And then what's the role of bees when they're transmitting pollen to pollen? They can, when they're doing pollination, are they transmitting viruses too? And people really haven't looked at that well.
Yeah. How are these transmitted among different bees?
Yeah, so all the typical ways. So we have vertical transmission from queen to egg, horizontal transmission, and this is, I always say this is honeybee colony is a great place for virus transmission. When you think about it, 40,000 individuals crawling all over each other. They do this thing called trophallaxis, which is basically mouth-to-mouth feeding, another excellent way to transmit viruses. And then between colonies, they, if one infected bee lands on a flower and gathers pollen, they may deposit virus onto that flower, and then another bee visits that and can we know that bee viruses are transmitted on pollen resources.
So is every colony in the U.S. or in the world infected to some extent, or are there some that are not?
Yeah, most colonies, even when we look at, we usually use colony population size as a proxy for colony health. And even if it's a healthy colony full of individuals, good honey stores, in all practical purposes, it looks healthy, we often see bee viruses. And I kind of liken that to, if you were going to take samples of people on the bus in the morning, and we analyzed what viruses were associated with them. Several would have rhinoviruses and other types of viruses, but maybe, asymptomatic. And so definitely we see that in bees as well.
So some of these viruses do not cause disease and others do then, right?
Yeah, and even the ones that cause disease, so deform wing virus is probably the best studied, and of course it causes deform wings, and that's easy to see. But even in bees that are asymptomatic, we can find billions of RNA copies, whether that's transcript or genome, we can't tell, with qPCR, but billions of copies of deform wing RNA in an individual bee. And so I think even if they're asymptomatic, to me, that seems at least they would be energetically expensive for the bee to be producing all that virus.
You mentioned the mite itself can be a vector for viruses. Can you tell us about that?
Yeah, the mite, aptly named Varroa destructor, can be both active vector or that actually harbors replication of virus, such as deform wing virus. But it also can be a passive vector where it basically just sucks up the bee blood or hemolymph, and then transmits that virus to the next bee that it bites, not unlike a tick or mosquito would do to human populations. But so it can passively transmit other viruses, and that's what we think it does for the Lake Sinai virus group. It doesn't seem that the Lake Sinai virus group replicates in the mites themselves.
So these are ectoparasites, these mites.
Yeah, they're
right?
ectoparasites. They say if you wanted to compare the size of them on a bee, it would be like the equivalent of a large dinner plate walking around on you, if we scaled. So they're pretty.
And they are
burdensome.
They kind of look like dinner plates too, right? They're these round,
they're flat red mites.
Which I guess is important for aerodynamics for them.
Yes, and for they get under the tergites of the bee, the little segmented body parts of the abdomen. They kind of bury themselves on in there. And there's new evidence from the University of Maryland, Dennis Van Ingelsdorp, that suggests that they burrow in a certain area where they actually consume the fat body of the bee, that's the kind of the organ of an insect that produces these hemocytes or immune cells. So it's a major immune organ of the bee. And so the mites eating that is, of course, not good for bee immune mechanisms or immune health.
They're really annoying.
Yeah, pretty bad.
So now when beekeepers encounter these, they, I don't know, you probably can't just grab the bee and pluck it off, but are there some treatments for them, like miticides to kill the mites but not the bees?
Yeah, and this is a really active area of research as well. But there are miticides, and commercial beekeepers in the U.S. if they were an operation that lasted through the onset or incoming of these varroa destructor mites, now all commercial beekeepers are treating for mites, and there's both organic treatments, which include things like formic acid patties that work as a fumigant, and then the mites fall off the bees, and the dose that's given is bad for the mites, but not bad for the bees. And then there's also synthetic miticides, which are which we worry then about resistance and things like that. And then how, although mites are arachnids and bees are insects, the miticides, they're harsh chemicals, and, you wonder what implications those have for the bees too. And so it's a hard thing to deal with the mites. The mites are worse for the bee colony, and you will inevitably have your bee colony die if it becomes infested with mites above levels of 4%. So we actually, this is another cool thing you can look on YouTube for. There's this powder sugar test for mites, and you put about 300 bees in a jar, and you have a screen lid on that jar, and you put a tablespoon of powdered sugar in, and you shake it up, and you shake that jar, invert the jar, and shake it over a pan of water, and then count how many mites fall off, because that powdered sugar gets in their legs and lets them release from the bees. And so we test for mites in our colonies once a month.
Okay.
So can you tell us a little bit about how you work on bees in a lab? Where are they, and how do you manipulate them and so forth?
Yeah, so we go out to our colonies, which are about 2 miles away from campus. We bring in a frame of and it looks just like honeycomb, except for instead of honey in there, you have developing bees. And bees take about 21 days to develop from egg to larva to pupa to a fully adult bee. Then the adult bee chews their way out of the cell, and that's my science pick of the week. You can actually watch this on a Nat Geo video. Watch the bee chew its way out of the cell, and then we call those newly emerged worker bees. And we use those for laboratory-based studies. And since the queen lays 1,000 eggs a day-ish, we can, reliably get 250 to 500 bees emerging in the lab, simultaneously. They're age-matched half-sisters. And then that's what we use for our laboratory-based studies.
Mm-hmm. And how would you infect them?
So we can either infect them orally. You can, put a Eppendorf tube that has little holes in it with a 50% sucrose syrup with a virus in there. But then you would have several bees feeding off that same, Eppendorf tube. And I don't like that as much, because then you're not getting to know that each of those bees in that cage got the same dose. So then actually you can do something more tedious, which is to put each little bee in the upside down Eppendorf tube, and you can get her to stick out her proboscis or bee tongue, and you give a 2 microliter dose of virus inside a sugar syrup. She'll smell that sugar syrup and stick out her tongue, and you can feed her 2 microliters. Or our favorite way.
So you're bottle feeding bees.
Yes. But we don't do that way very much, because, it does, as it sounds, it is tedious. And so the more common way is that we give them artificial mite bites. We use hand-pulled capillary needles, and we give them a dose of virus using a modified syringe and these glass pulled needles. And we give them a bite in the side, which is a 2 microliter dose of virus at a specific titer. And then they survive that injection very well. Actually, we lose less than 2% of our bees in this injection process. And then we control the dose of that virus, which is really important. And
Where do you inject them? Is it in the abdomen or?
No, the abdomen is squishy. We tried all this stuff. The abdomen is too squishy for these needles, and we typically go right under the wing. There's a little soft spot. It would kind of be like the armpit of the bee. That's where we inject them.
And how do you house the bees in the lab?
So yeah, we make these modified deli containers, complete with a jungle gym. They have a little piece of plastic that has beeswax coated on it, so they can climb up and down that. We give them some enrichment. I think it's what's called in when you have mouse colonies. And we have them in these deli containers where there's a sponge with water to have them have water, and then increase the humidity in that chamber. And then we can feed them a little bit of pollen supplement, which is their protein source, as well as sucrose. 50% sucrose sugar. And then those are nice, because then we can just autoclave and be done with those containers after every experiment.
So how often do you get stung?
Yeah.
So I get stung very rarely. I wear all the gear. I wear the gloves, and I make all the students wear all that stuff. And so you can definitely tell we're scientists and not beekeepers, because beekeepers typically work bees without gloves. They'll maybe have a veil, maybe not. But I only get stung, once or twice a year, and it's definitely always my fault. If you don't wear proper shoes and you drop some bees in your shoe and then you step on them, they're going to sting you. But in general, they're very peaceful. And so we bring student groups out to the colonies every year. They wear veils, but I also constructed this pavilion that you can pull a screen all the way around, like a screened-in porch. I think people in the South have them. Maybe Rich has them in Florida, where we pull the screen around the pavilion, and then they can be inside the screened walled-off area, and I can bring frames of bees up to them, and they can be inches away from them, but in no danger at all. And that's really cool. To watch the kids be so fascinated by the bee colony and to get to observe how peaceful they really are. I mean, when you think about it, you've pulled out, members of their colony into the lights, and as long as you move calmly and slowly, they just go about their business. You
mentioned in your paper something called bee candy. Is that the stuff you put in their deli containers?
Yeah, we mix up, and it doesn't sound like a very good diet, but at least we know it doesn't have confounding pathogens in it. It's corn syrup and confectioner's sugar, and mix that up pretty thick. And it's funny, I think I studied to be a microbiologist and virologist, and all this stuff was empirically determined as well. If you make that sugar candy too wet, the bees can get stuck in there and then they die, and then that ruins your experiment. So all these little details had to be worked out.
And what's bee bread?
Bee bread is what they store in the colony, and that's when they store pollen inside what looks like honeycomb, but then instead of nectar that's dried down to make honey, it's pollen, and it can be a variety of colors. It's really beautiful to look at, actually. And then there's some microbes in there, some fungi and bacteria that potentially break down some of that pollen, and then they call that mixture of the microbes and the pollen that's stored, they call that bee bread.
Mm-hmm. I wanted to ask you about a cell line. You mentioned, I think you sent us a review in Current Opinion in Insect Science, and you mentioned that there's the first transform bee cell line. Is that right?
Right. A first, cell line that was basically, it was a mutated cell line, because then it, became immortalized on its own it originated as an embryonic stem cell culture.
I see. So it was subjected to mutagenesis, or it just happened?
I don't think so. I think just by chance, just by propagation. But unfortunately, that cell line isn't that reliable, and it's difficult to keep in the lab. We've had Michael Gorblish, who developed the cell line at University of Minnesota, and Marla Spivak's lab out twice to MSU, and we have the cells sitting there, but they don't, they take months to divide. They're just not like as easy as working with HeLa cells, which would be great, or some other kind of rapidly dividing cells, or even Drosophila S2 cells, which we also have in the lab.
Right. 2 months
to divide. Probably just as well, because then I know we're going to be talking about the antiviral responses in bees, and if you were also working with bee cells, that would get very confusing.
Exactly.
So it would take 2 months for these cells to divide?
Yeah. And so then it's just not, imagine my PhD students are, so that's why Alex goes to the, kind of lines out a bunch of experiments for the summer, obtains hemocytes or primary cells from larvae, and then uses those, because that's just more practical.
Right. So that if you want to use a honeybee cell, you use hemocytes, because there's nothing else available at the moment, right?
Yep, that's correct.
Although, so you mentioned the S2, that's a Drosophila cell line. Do you think there'll ever be something equivalent from bees?
Yes, I think so.
People are working on it. Okay.
Yeah, I think a lot of people have worked on it. It's one of those projects, especially as a relatively new faculty member, you can't make that someone's PhD project, because if it doesn't work, it doesn't work. But I think everybody is trying it, and I just hope that something works out at one time. I mean, people are propagating cells and trying to make cell lines, but nothing's come yet.
Yeah. Well, and the fact that there is one, it's just very, 2 months dividing time and all that. That's it suggests this can be done. It's just a question of getting the conditions right.
Yeah.
Let's talk about your paper you sent, Scientific Reports. Virus and double-stranded RNA-triggered transcriptional responses reveal key components of honeybee antiviral defense. So you have 3 authors. You got yourself and 2 other people, Laura Brucher and Katie Dochenbach. What are they in your lab? What's their positions?
So Laura Brucher graduated with a PhD from my lab. She's my first PhD student,
Cool.
and she's now a postdoc. Thank you. She's now a postdoc at UC Davis in Alina Nino Lastro's lab. And then Katie Dochenbach, I'm super fortunate to have. She's a PhD virologist. She did work with Norwalk virus, and she was luckily coming back to work after having 2 kids when I was coming back to Montana State. And I'm really fortunate that she works part-time in my lab. She's an expert virologist and a great lab manager as well.
Great. So tell us the backstory of this paper.
So this work originally started when I was a postdoc in Raul Landino's lab. And at that time, and as he's been on TWiV describing, in Drosophila melanogaster, it's appreciated that RNA interference, that sequence-specific post-transcriptional gene silencing or virus silencing mechanism is critical and the major antiviral response in fruit flies. And so while in Raul's lab, I wanted to see, well, is RNAi the major antiviral response in honeybees? And so I did parallel experiments in fruit flies and in honeybees where we co-injected virus and double-stranded RNA that was specific to the virus. So that would result in an RNA interference response. And then, of course, the control, which is double-stranded RNA that has no viral specificity. It either is specific for luciferase or Drosophila C virus or something else. And then that was supposed to be the control. And I always say that this is, just a good commercial for doing all your controls, is that unlike what is seen in Drosophila, what we see in honeybees is we do see, and others have shown, that RNA interference, which is triggered by sequence-specific double-stranded RNA, is important in bees. Double-stranded RNA in its own right triggers an immune response in bees. And I think to mammalian virologists, we're all thinking, oh yeah, that makes sense. Of course it would. We think of the interferon response in mammals as a major antiviral response. But this was a weird result for insects at first because it had never been described that double-stranded RNA, regardless of sequence specificity, reduces viral abundance in honeybees.
But there's also a Argonaut dicer-based system in honeybees as well?
Yeah, and I think if we hypothesize, and of course need more data to look at this, is that, when you think about a fruit fly, it's a solitary insect. It needs to be ready to just fend off viruses on it in its own right. And maybe RNAi then has evolved to become the major antiviral defense. And we already talked about how honeybees live in these colonies of 40,000 individuals, part of this superorganism, great places for viral transmission. And so maybe honeybees have both this RNAi or virus-specific defense mechanism, but also, like humans, have this generalized antiviral response that's just going to trigger some antiviral responses to any virus once stimulated.
So what did you do in this paper to look at this?
So we had newly emerged bees in the lab, and we injected them with a model virus, in this case, a Symbis virus tagged with green fluorescent protein. And we either co-injected double-stranded RNA that was specific or non-specific for the virus. And then we monitored viral abundance over time where these bees were housed in the lab. And we took samples at 6, 48, and 72 hours post-infection. And we examined viral load and found out that double-stranded RNA reduced viral load, as well as poly-IC, the synthetic mimic of double-stranded RNA. So that was both sequence and non-sequence specific. We chose to focus on the non-sequence specific aspect of this because it's different and most interesting. And then we did a transcriptome analysis, and we did this in individual bees. Because, as I've mentioned a couple of times, but really haven't explained, the bees in our studies are only half sisters. So they're not like Balbc mice clonal. Right. They are only a queen bee mates with several drone bees, up to 10 to 12 male bees. And so the bees that are merged in one colony are only half sisters. So they get half their genetics from the queen and half the genetics from the male bees that she mated with, but she mated with, 10 to 12 bees. And interestingly, as a side story, fertilized eggs in bees become female worker bees, and unfertilized eggs are drone or male bees. We took these half sisters and we had to do we did transcriptomics to look at the genes that were turned on in the context of both virus alone, virus plus double-stranded RNA, as well as double-stranded RNA alone. And we again did that on individual bees that we had already screened for confounding infections. And we found over the course of infections, hundreds of differentially expressed genes. We found a major finding was that dicer and argonaut were exhibited increased expression in the context of viral infection, and that's actually different than Drosophila. These proteins are not regulated at the transcriptional level in Drosophila, so that was also interesting to figure out that components of the RNAi pathway were transcriptionally regulated. And then we identified new genes that are important antiviral effectors, and we're really pursuing what are the functions of these genes. And one is this, we called it a putative cyclin-dependent kinase in this paper that Laura wrote, but we don't really think it functions as a kinase any longer. Further research doesn't really support that. And so this gene, which we now just refer to as sporine name, is MF117363, is this new transcript that we actually annotated. It wasn't annotated well in the honeybee genome. And we think it's really important for antiviral defense because when we knocked it down in bees using an RNAi approach, if you knock down this expression of this gene and infect those bees with a virus, they have higher viral titers than the controls.
So when you do that, when you say an RNAi approach, you inject, what do you inject into the bees to do that?
Then we inject double-stranded RNA that's specific to the gene.
Right.
And of course, this is an imperfect experiment, but as we kind of already have described, it's a challenging system, but important system to study. And so since we can't there's not a lot of genetic tools for bees, like other non-model systems, injecting double-stranded RNA to reduce gene expression is an important tool.
So you could target any gene in the bee by injecting a specific double-stranded RNA, right? And then the mechanism involves argonaut, the knockdown of that target?
Yes.
Okay. And that's different from what you think is going on when you put in non-specific double-stranded RNA, right?
Yeah, and so even in Drosophila, they've shown that dicer, which is a dead box RNA helicase protein, it can also function. It's the same broader family as RIG-I, MDA5, LGP2, and other double-stranded RNA sensors that are in the cytosol of cells, and so that when they bind double-stranded RNA, they can potentially interact with other proteins and kind of result in a signal transduction cascade that results in the activation of immune response that's not just the risk-mediated slicing, and argonaut's the slicer in that case, the risk-mediated degradation of virus.
Right.
We think that there's parallel to the interferon system, there are hundreds of different genes that are turned on in response to virus infection.
So, okay, now I know insect immunity has not been terribly well studied, and Drosophila obviously has gotten a lot of the attention. And so in Drosophila, we've got the RNAi system, which is specific to particular viruses. In humans, obviously, we have double-stranded RNA responses through interferon. What you found in bees is they have this double-stranded RNA generalized response, generalized immune stimulation response. And it's using, if I'm understanding this correctly, mostly genes that don't look like the stuff that's going on in humans. And this isn't a system that's active in fruit flies, right?
Right. And so beyond that, these genes, a lot of them have double-stranded RNA binding domains. We don't know what they do. And this is really interesting. If you think, although the honeybee genome is sequenced and we estimate there are 16,000 genes encoded in the genome. Only about a third of them have, well-studied orthologs in fruit flies or mammals. And so a lot of the genes that we identified, you get this hit back that just says LOS, this, transcript of not been described. And that's the case for one of the main genes in this paper, that putative cyclin-dependent kinase that I described earlier was just labeled this kind of RNA.
So that suggests that this is a system of that we've had parallel evolution here, where this double-stranded RNA response evolved in honeybees, perhaps, as you suggested, because they need it to protect the colony. And then millions of years later, in our own line, in the in more complex organisms, we get an evolution of a double-stranded RNA generic response for our own reasons, because we're evolving a more complex immune system. And we end up using stuff that obviously is very evolutionarily diverged from what the bees ended up using.
Yeah, it's interesting though, because, of course we just have these, the transcripts that then we can, estimate. We don't have structures. I mean, there could be a lot more parallels in these, just because I blast them and it doesn't come back as LGP2. There could be, a bee protein that's very similar to LGP2 or RIGI or MDAA5. We just don't. It just doesn't look like it, and we would need structure work to show that or something like that.
Right, and either result would be really interesting, because if this is evolving, if this evolved in parallel, completely independently, that's cool.
Yeah.
But if you've got RIGI in bees and a homologue of RIGI at the protein level in bees, that would imply that this is a much, much older system than we previously believed.
Yeah, and I think both are really fascinating, and we would, of course, love any genes or mechanisms that we identify in honeybees. We would be happy to move that investigation into mammalian cells and see, are there parallels or,
Right.
And another interesting thing is that, and I used to think about it backwards till I reminded myself that when you look at insect evolution, the Hymenoptera, which is where honeybees are, that includes honeybees and wasps, they are more basal to the tree of life than Diptera, which is Drosophila. Right. So then Drosophila evolved later, maybe 100 more million years later. So maybe they had both and then lost this arm because they didn't need it, or RNAi was so much better.
And maybe this has some similarities to parts of the RNA innate immune response that we don't know about yet in mammals.
Right.
Yes.
I want to ask a little, I know I read it and maybe you said it, why you needed to use the Synbis virus. And did you already know that it would work in bees? Because I think of it as being a mosquito virus.
Yeah, no, and it is a mosquito virus. I didn't know it would work in bees, and I still remember the day when I saw the glowing bees under the microscope. Actually, Raul mentioned Pat O'Farrell at UCSF, and it was in his lab using his microscope, the same one that Carla Soleil used, and Michelle Tizzetto that were postdocs at the time. And so I was really delighted to know that this insect virus that was tagged, I think in Charlie Rice's lab actually, could replicate in bees, because now we had a system. I mentioned that in our other studies, we purify virus from infected bees, and that's an imperfect process, although we have gotten pretty well good at it for the Lake Sinai virus group. And then we use those to infect bees. But this system is just really avoids working with confounding pre-existing conditions, we call them, that you can't know about.
And you don't have an infectious clone for any of the bee viruses. So This is a way to deal with that.
Yeah, we have Benjamin Lamp made one, and we have that one in the lab for Deform wing virus. And then we're working on one for Lake Sinai virus too. But it's not there yet.
Mm-hmm.
So as an infectious clone, does that mean you can inject the DNA into the bees and get virus replication?
I think what we do, and we've tried, is to inject the RNA in. We can make do in vitro transcription, inject the RNA, just similar to polio virus.
But a great example.
Yes.
So you say in the paper there are currently no honeybee virus isolates. So by that you mean you don't have something that you've grown up in a cell culture and is clonal and so forth, right? You only have ground up bees, and you don't want to use these for confounding reasons, right?
Yeah, and we do both. So we do studies, and other labs as well do use both. But flockhouse virus is another model virus that we have in the lab, and there's been some cool work on that by Scott O'Neill, showing that this anion channel was important for to defend against viruses. Probably there's like a link between metabolism and this potassium channel. And so flockhouse virus is another one that's used. Deform wing virus is really acute paralysis virus, but the deform wing and IAPV that have been used have always been purified from pupa. And I say purified with those air quotes around it because it's hard to purify.
Okay. So what is more important for the honeybee, the specific or the non-specific double-stranded RNA response? Do you know?
No, I don't know. I think I would just answer both, but I don't know. I don't know the relative importance of those systems. Is there some way to get at that at some point? Yeah, I think so. You could target multiple genes in one pathway and then both pathways simultaneously in the bee. Again, though, this is depends on being able to effectively knock down multiple genes in a hypothesized pathway or divergent pathways. And as we all know that there's different mRNA half-lives and all these things. So again, imperfect experiments, but I think really fascinating and worth doing.
But you could do a dicer knockdown, right?
And we did that.
And
we did that in this paper. We knocked down dicer and we saw that virus was increased as compared to those bees that just had double-stranded RNA on its own.
Right. And then your cyclin-dependent kinase, you knock that down and you also get an increase, right?
Yeah. And so this summer we'll knock down both at the same time.
Okay.
And then look at that.
So,
and I guess you can't really go into the Drosophila world is full of mutants and knockouts and all sorts of genetic modification. But with bees, you would have to then raise them in a hive, and I'm just going to hazard a guess that genetically modified bees in a field release at the laboratory would probably not be popular, right?
Yeah, that would probably shut down my career pretty fast here. But people are doing so. CRISPR-Cas modification of honeybee genome has been, I think, shown by one group in Japan. I know Leonard Foster's group at UBC in Canada is working on it, but of course these are very contained just for experimental purposes of understanding what's the function of a particular protein in a bee. And you can imagine they're very technically challenged because you have to take those little newly fertilized eggs or just tiny embryos, inject them in the lab, and then actually have them develop into this organism, which is definitely not trivial, and then have enough of those that actually have your knockout to study. And so although probably feasible in the future, it's not something that we're trying in the lab right now.
So and you can't read the knockout in the lab.
No, you could actually, if you had, so that's something cool. I mean, you could, if you raise drones, which would be then haploid, you could raise them and then you could, and they actually do this, and I've tried this, I'm not very good at it. They do, just like in other egg, they do artificial insemination of honeybee queens or instrumental insemination. You can actually do that. And so you could, the techniques are developed that you could have a singly mated honeybee queen that then you could raise, sisters, not just half sisters, and study them.
I think that's what they do with ants, right? They have ways to make them independent of the queen. In fact, we had an ant TWiV not too long ago where they were able to make the first knockout because of that. Because you can't keep going back to the queen, obviously, because then you're going to lose your knockout. But somehow there's a way to propagate the workers.
And then for honeybees anyway, you could propagate workers actually, and this happens in a normal natural honeybee colony. If the queen dies and there's no queen cell or replacement queen, then you'll get what's called laying workers, and they'll lay a bunch of eggs that will be unfertilized, and then they're unfertilized and then will develop into male bees. And so I'm sure there's ways that you could kind of trick colonies to do what you need to do.
Is that something that you're interested in, or?
I think down the road, I think pre-tenure here at Montana State University, I got to stick to some more rapid experimental findings.
Yeah. But I mean, certainly it would be nice to have a bunch of mutants, as in Drosophila, with your bees, right?
Yeah, and originally, when I first got into this, I thought, well, we'd always just go back to Drosophila. I had to order those mutants from Bloomington Labs and study those genes. It just is in the pathway I'm most fascinated in now. The Drosophila and the bees are different, and so
it sounds like the flies. Sounds like flies and bees are different enough so that you want to be really careful there.
Yeah, that's what I think too.
Yeah, I hadn't appreciated all these challenges and difficulties. I thought, just make libraries of mutant bees, but it's not that easy. Yeah, that's
so it could be something like bees lie and Drosophila exaggerate.
Yeah, that's right. Now, we're talking about Drosophila and honeybees. Does anybody do any of this with other bees or other insects or arthropods?
Well, interestingly, a group in Belgium, Guy Smaggy's group in Belgium, studied double-stranded RNA and RNAi in bumblebees, and I was heartened to know, because you can imagine, when you get these results that are first different than Drosophila, and then they're, it's an imperfect system. He also observed that double-stranded RNA in its own right, regardless of sequence specificity, reduces viral infection in bumblebees. And so, bumblebees, like honeybees, are Hymenoptera. So that's kind of
that's
cool.
Yeah.
Yeah.
But they're solitary, aren't they?
Well, that's interesting. Really interesting. So they're sometimes solitary, like when they first emerge in the spring and you see these huge bumblebees, they are solitary, and that's the queen bee. But then over the course of the summer, she raises, colonies that can be hundreds of bumblebees, and then she stays back in the colony, and then the worker bees are out foraging. And so bumblebees are really a cool example of sometimes solitary and sometimes eusocial insects.
Neat.
Now, your bees that you raise in the way that you told us, do they have other virus infections? And could that be an issue?
Yeah, it can be an issue. And so before doing RNA-seq analysis, Laura had to do, many, many different reps, biological reps with bees obtained from lots of different colonies. And then she screened by qPCR, I think probably for 12 different common viral pathogens, as well as trypanosomatid pathogen and fungal pathogen. And then we sent for sequencing the cleanest biological rep that we had that didn't have appreciable levels of confounding infections.
Yeah.
But that's a problem studying something that needs to live in the environment.
Yeah. That's another reason why you'd like to put it all in the lab, like Drosophila, right?
Yeah. And some labs have flight rooms where they keep a colony of bees inside. They do that at Iowa State University, I think in Brione Bonning's lab. And I visited Francesco Nassi's lab in Udine, Italy, and they have a flight room. But in general, when you look at those colonies and you look at the room, it doesn't look like that healthy of a colony. So if you're studying immune responses, it just seems like I haven't ventured into this flight room, because then I think I'm having other confounding problems.
Yeah.
Do these flight rooms have plants for the bees?
No, usually they don't, because then if you're doing that level of effort, you're trying to really minimize. So you'd have artificial diets that would be sure not to have any chemical contamination or pathogen contamination.
So really an intermediate system would be a completely contained greenhouse, I guess.
Yeah.
And that's how they determine. So Diana Cox Foster's group at Penn State University and others working with her to use a greenhouse study to show that Israeli acute paralysis virus could be transmitted between honeybees and bumblebees in a greenhouse setting.
Okay.
And those are sealed so the bees don't go out, right? Wow.
And then if you were doing that, so if you provided in this, I also study potato virus Y and especially in potatoes, provided we use a local isolate of a virus that's already circulating in our area, then you could, in Penn State, they use the IAPV from Pennsylvania, and most colonies test positive for IAPV. So, it's not such a biocontainment concern if you're using a virus that's already circulating.
I want to just ask about all these hits that you got in this paper. What's the plan going forward? You can't work on all of them. Are you going to focus on this cyclin-dependent kinase?
Yeah, we're focusing on that. We're also focusing on the heat shock response, because that can be both pro and antiviral, and that's very interesting. And some of the other genes, a handful of them we're focusing on and trying to drill down. What's the sensor of the double-stranded RNA, things like that. But of course, we can't study all hundreds of these genes at once.
Yeah, that's an interesting question. I was thinking of the same. What's the sensor for? This is for the non-specific double-stranded RNA.
Yeah.
Right?
Yeah, and we have a good indication on that. We're doing, some RNA pull down works with an antibody that binds double-stranded RNA, and that's Alex's project, and hopefully he'll have some answers for us in the next year or so.
But there are no homologs of mammalian sensors, right?
No, the only part that you get is you get domains. So you'll get,
sure.
Yeah. So then, I'm not that well versed on where you'd call it, the cutoff or not, but you'll get domains,
right,
but then not the whole protein.
Go ahead, Kathy.
Oh, I was just going to say, this paper is open access, and I really liked Figure 3, the visual for the results that you got. They're simple Venn diagrams, and then they point to the table. It's really visually presents a whole lot of information, and it's easy to take in. And then, Figure 6 is the funniest looking Venn diagram I've ever seen.
Yeah. It was funny. I agree.
People just need to take a look at it.
I really wanted to do it, and it just compiles all the different transcriptome studies that had been done at the time, and it's hilarious. But I really wanted to know what were the we identified that were the same as others, and what were different.
Just to tell everybody that each kind of virus has kind of a shmoo-shaped overall thing, and then the narrower shmoo parts of them are kind of overlapping and make this inner star, and it's just amazing.
When I first downloaded the paper and flipped through it, I was like, oh, and there's a picture of a flower at the
end. All right. Anything else? Did we exhaust honeybees?
No, there is so much more you can learn about honeybees, but I know that not everybody wants to hear about them all day, and so I think we covered most of the main stuff. And another main question I get is, what can people do to help bees? And in general, I just say use less insecticides in your own yards or gardens, or buy local honey, or plant flowers, which are all easy and things that people can do to help bees.
Mm.
So we're going to talk about these videos and and the outreach program. Okay, good.
Yeah. Tell us. So you said links to these cool videos. Tell us all about that. It's based on your NSF grant, right?
Yeah, so I have a NSF career grant to look at the immune mechanisms of honeybees, but as part of that, there's these broader impacts, and I really am a firm believer in scientific outreach and do a lot of it. And so I interact with student groups, Native American high school groups throughout the summer, 900 fourth graders every year in 15-minute intervals at this thing called the Gallatin Valley Farm Fair. So I was finding myself exhausted from these efforts. And so we decided to make a short video series that teachers, or just interested individuals could look at and get a sense of those kind of classes that we do at MSU, or seminars that we do at MSU for different learners of all ages, really. And so we made a video about honeybees, why they're important, honeybee pathogens, and then really even how we detect honeybee pathogens using molecular techniques like PCR and quantitative PCR. Because actually, beekeepers get viral test results. There's a project called the Bee Informed project, and you can send in bee samples and get your virus. Viruses from your bees. And then they're really, if you haven't had science classes or genetics in a while, like many beekeepers, then those results are confusing. How did they get this? I have 10 billion black queen cell viruses in my colony, and should I be concerned? I get calls like that. And so it's kind of good to explain how we detect pathogens to beekeepers as well. That's why we made the videos. It was pretty fun to do. The MSU communications group worked with us, and they were fun.
They're nicely done.
Very well.
Yeah, they're
done.
very well done.
Very well.
I really like watching the students in the videos.
Yeah.
See how engaged they are.
Yeah, they really like it. I've had, just been walking down the street, and like we mentioned, Bozeman's a small town already, and some kiddo will just run across and hug me on the sidewalk because I was their teacher over the summer, and the parents are kind of looking like, who is this lady? And she's like, I learned about bees from her.
She's the bee lady.
Yeah, exactly. They call me that. We do talks at the museum and talks at this old school theater downtown called the Ellen. We have MSU science roadshows, and definitely I have been called the bee lady inside a grocery store here.
Awesome.
Well, we'll put links to these videos so everybody could look at them. They're pretty. I'm just impressed at the quality. Really well done. Yeah. It's not easy to do that.
Good video is hard.
Thank you.
It is.
All right, let's finish up with some picks of the week. let's start with you, Michelle. What do you have for us?
I found this really hard to just pick a single pick. So I have several, and I could have kept going. But one of the picks of the week was this Anand Verma TED Talk, the first 21 days of a bee's life. And it's amazing footage. He worked with a beekeeper out in Berkeley, California, and looks at bee development from an egg to the newly emerged bees that we talked about today. So that's amazing.
This one is really cool. And it's don't be put off by the idea that it's a TED Talk. It's only 6 minutes long, and it's so worth your time to go look at.
It's really good.
Yeah, even the fourth graders can watch this one. So that your attention span can be limited, and you'll enjoy this video. And then I also am really fascinated and interested in NPR's Hidden Brain podcast. So I thought that was a good one to pick. Something I listen to a lot. And then the last one was a bioarchive paper. We didn't really spend so much time talking about it, but a really interesting part of studying bee viruses is this mite deform wing host immune response complex, this like tri interaction, and what the consequences are of that for bee health and then viral development. And so this bioarchive paper takes another look at this and kind of talks about the reduction in immune cells from the mites basically drinking the bee blood, and then how that has a consequence on viral replication. So it's just an interesting way to think about it. And this is the last author is Francesco Nazzi at the University in Udine, Italy, and I had a chance to visit his lab last summer, and I think he's an excellent scientist, and so I chose this bioarchive paper.
That's not Nazi with two Z's.
Yep.
It's in Italian.
Sorry.
Yeah,
It's an unfortunate last name, I would say.
Yes.
Nazi.
What's the size roughly of the honeybee research community?
I don't know. Maybe it is, 50 or 100. It's not so many people. I feel like you do get to know each other, and it's definitely a really collegial group, especially if you drill it down to how many people study honeybee viruses, then we're getting really low numbers, less than 50 individuals. And so, so we mostly know each other.
Okay. All right. Brianne, what do you have for us?
Since flu season is on its way, I was taking a look at something called Flu Near You, which is a partnership between Boston Children's Hospital and the CDC and a few other groups. It's been recently featured on Science Friday, where people can be reporting on flu symptoms and using that to help local health departments track flu in their area. And so they have some pretty nice data showing the flu epidemics in different locations. And as we get closer to, or as we get into, later in the winter and we see more flu cases, this might be a very useful way for those of us who spend a lot of time with, say, students to know when to be worried about the flu.
Yes.
Alan, what do you have?
I have a link to a program. It's an outreach program called Skype a Scientist. And this is pretty much what it sounds like. So if you're a teacher, you can go and sign up as a teacher. If you're a scientist, you can go and sign up as a scientist. And it matches classrooms with scientists. And then you connect on Skype and you do a 30 to 60 minute question and answer session. Students ask you about your research and you talk about it.
Cool.
Excellent.
That's really cool.
Yeah, that's nice.
I did Michelle, I did a Skype class with a class at Bozeman.
Oh, yeah. You can do it again. I'm in charge of that.
Oh, Michelle Hardy used to do it, right?
Yeah, and we do that same thing. This year we did do, Sam Sternberg was one of our guests this year. We do Skype different scientists as part of our virology class. So next year, yes,
cool.
Love to have you again.
Yeah, Sam.
Yeah, this is aimed more at like elementary age.
No, this is our 400 level virology class. And so
Cool. Right.
No, yours is different, but I was just remembering that I did it with a class in Bozeman. I'm thinking, what? Michelle wasn't doing that, and then I realized it was the other Michelle.
That was a Skype and advanced virology class.
Yeah. That was a lot of fun. Rich, what do you have?
This has been in the news. Voyager 2 has now exited the solar system and it left the building and is in interstellar space. So this is Voyager 1 did this several months or a couple of years ago. I forget quite. But these are 2 space probes that were launched over 40 years ago for the purpose of interplanetary studies and ultimately perhaps leaving the solar system. And they weren't designed to last this long, but they did. This guy's still got at least 3 instruments working and sending back data, and he is now beyond the solar system, 11 billion miles from the sun.
It's
and
amazing.
won't be seen again until the Star Trek movie.
That's right.
Amazing.
So the article is pretty good, and it's a NASA article, and it's got a nice video about the whole thing and links to the Voyager program as well.
Cool.
Kathy, what do you have?
Well, I have a couple things before my pick. I checked, and TWiV number 1 was an hour and 31 minutes.
Oh, was
long.
it?
So
Okay.
I'm not sure we ever had
Somehow
30
I thought,
minutes.
Somehow I thought there were some
Yeah.
shorter early ones.
Yeah.
Oh well. By the way, episodes 2 and 3 are each like 45 minutes long.
Okay. Okay. And then for my picks of the week, I have never really had it very clear in my head what's a honeybee or what's a bumblebee, and they sound to me as generic as saying germs. So I decided I really needed to figure out what they were. So I went on the web and found some images that really compare them very nicely. And I ran into 2 things from the New York Times, both of which I remember seeing when they were in the paper. One is a quiz with a picture of a bunch of insects, and you're supposed to try and test yourself and figure out which ones are the bees. And another one is what it feels like to be a bee. And if you listen to this episode, you'll get at least one of the questions on one of those quizzes right, because Michelle gave us an important number. But I found those both kind of fun to redo today, and so you might like those.
Yeah, that's a great one, because I often get people telling me about how honeybees bother them at their picnics, and I say, unless you're eating nectar or pollen, I think those are wasps that are bothering you at your picnic.
Yeah,
people do mix them up. That's right. All right, I have 2 picks. One is pretty short. One is a pick in Scientific Reports, which is an open access journal, so everyone can go look at this. And it's a paper called Methylation-Based Enrichment Facilitates Low-Cost, Non-Invasive Genomic Scale Sequencing of Populations from Feces. So it's all about, you have some animals, you want to speciate them or make trees, and you can easily get DNA from feces. It's kind of interesting, but usually I don't pick a paper on TWiV, but the reason I pick this is because I want you to go to Figure 1.
I saw this on Twitter now that I see the paper.
Yes.
And blow it up and focus on the piece of feces next to the monkey. And it's a little Easter egg in there. Let's put it that way. Just really amazing for a nature journal. And I don't know if it's going to be there forever, but
I saw a message on Twitter today that made me think it's not going to be there forever.
So I have the PDF. If you don't see anything, if you don't see an Easter egg in the feces, I can share the PDF with you, but it's kind of funny. It's kind of unprecedented, I would say.
Yeah. So there's an editor's note on the website right now that says the editors have become aware of unusual aspects to the extract fecal DNA illustration in Figure 1. We are investigating, and appropriate editorial action will be taken once the matter is resolved.
Yeah, well,
we're going to mess up. Yeah,
they're gonna censor it, I'm sure. But it's interesting that by the time this is released, you might not see it. But it's just interesting that it got in there and so forth. But so that's cool anyway. And then my other pick is Peter Hotez's new book, which is called Vaccines Did Not Cause Rachel's Autism, or subtitled My Journey as a Vaccine Scientist, Pediatrician, and Autism Dad. So some of you may know Peter Hotez from Baylor College of Medicine, has worked on a variety of parasite vaccines over the years. We've had him on TWiP years ago, interested in neglected tropical diseases, and happens to be a pediatrician as well, and had many years ago a daughter diagnosed with autism. And this is a book about his experience. I mean, he's really trying to counter the anti-vaccine movement, which in part is rotating around vaccines causing autism, which of course is not true and has been disproven, but which continues to persist.
It's amazing that won't go away. Just amazing.
And, he said today there was a long period between the publication of the original Wakefield paper and the retraction by the journal. And he said in that time, all the damage was done and it persists. And so this book is about that. It's about the vaccine, the anti-vax movement, the focus on autism. And then there's a lot of personal stuff in it about his daughter and what we know about autism, all the genes that have been identified. They actually had his daughter's exome sequenced, and they discovered another gene associated with autism. But, the message is that vaccines do not cause autism. Here's my personal story, and from a scientist and a dad. So it's really good. And I spoke briefly with Peter today, a 20-minute TWiV, which I'll release next week, and you can hear him talking about it as well.
That's great. We always assign books for extra credit, like Paul Offis's Vaccinated or David Quammen's Spillover. And so next year we'll assign this one. I think sounds like a really great book.
Yeah, it's really good. And it's his when you make it personal, as we've been told in the vaccine era, it's sometimes has more weight, right? Instead of reciting facts, just say, well, this is my experience. And so Peter said he wanted to do this, and, he follows in the footsteps of Paul Offit and others. And he said he's already getting lots of hate mail and so forth. It's just been out a month or so. So that's too bad. But, he really believes that we all have to do something to combat this. I was on the way to Europe a week or two ago, and there were signs in the airport that says there's a measles outbreak in Europe. Make sure you're vaccinated. This is ridiculous. It's completely vaccine preventable. Should not be happening.
Yeah.
All right. So that is TWiV 525. You probably listen on an app on your phone or tablet. Please subscribe. I know some of you like to just go to a computer and play it, but it really helps us a lot if you subscribe. You get every episode that contributes to our download numbers. So please do that. We love to get questions and comments. TWiV@microbe.tv. If you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a couple of ways that you can do that. Our guest today is from Montana State University in Bozeman, Montana. Michelle Flenniken, thanks so much for joining us.
Thanks for having me. It was really fun.
Lovely learning about honeybees, all sorts of things I didn't know. It's great. Brittany Barker is from Drew University. She's on Twitter, BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here. Are you sure you don't have anything else for me to do instead of grade?
I know you were grading the entire podcast. Did you get it done?
I was not grading the entire podcast, so alas, I am not done.
Well, right. You've been just doing this online for the past couple of hours, so you really ought to go out on Twitter and make sure that everything's updated there and maintained properly.
I probably do need to do a few things like that. You're right.
You're teaching again next semester, right?
I'm teaching next semester starting on January 14th, but I'm also taking students on a study abroad course starting on December 27th.
Excellent.
Where are you going?
We're going to Johannesburg, Durban, and Cape Town, South Africa.
Wow.
So that's for like 2 weeks, 3 weeks?
So we get back on the 13th, and spring classes start on the 14th.
Wow.
Cool.
All right. Yeah. Have fun.
I will. It's going to be great.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's at turbidplaque.com. You can find him on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.